I'm talking to the class. Thank you. Good morning, everybody. So welcome to the continuation of our lectures on the cell biology portion of the cell biology biochemistry course. We're going to go through these two, and pretty much tomorrow with the next two we'll be done with the entire portion of cell bio. Then you will be focusing more on biochemistry, genetics, going a little bit deeper into stuff. So today we're going to talk about, I would say, interesting treat for you in a sense, is we're going to discuss a very important function of the cells. We're going to discuss cell signaling. We already mentioned that cells are in constant communication with each other, and cells are also in constant communication with extracellular matrix, and hormones and other structures secreted somewhere in the body will influence the cells. So this is achieved through a process of cell signal. This material is very important for you. Why? Because it's very important for understanding of future pharmacology, physiology, even biochem. So it's definitely something that's also highly tested on your board exam. So let's start talking about the first group of signaling, molecule signaling proteins that are known as G proteins. So these are intracellular signaling proteins, and they're named G proteins not because they're associated with government, but because they actually are named by their ability to bind to guanosine triphosphate. And of course, then they have a capability to hydrolyze GTP into GDP and release phosphate. Now, there are two families that we're going to talk about today in detail. First one is RAS superfamily. And, RAS superfamily almost sound like Rastafarian superfamily. This guy going to remember it, It's a good thing, Jamaica and everything else. So what it is that RAS superfamily are actually signaling G proteins that are monomers. They share just one unit. They're very similar to this one subunit monomer of heterotrimeric G proteins, the other group. And they will receive signals from catalytic receptors, which we're going to mention in a moment. They're on the surface of the cells, and they will be activated by binding to a ligand. Now, what's a characteristic of these RAS proteins? They're involved in signaling that regulates cell proliferation, how much cell multiplies, and cell differentiation. How does the cell converts how does the cell being pushed towards specific function? The next one, heterotrimeric G proteins. That means they have 3 subunits. And these are, I think the majority of the G proteins are heterotrimeric G proteins. So they have 3 subunits. They have alpha subunit, beta, and gamma subunit. And now we're going to talk about the G protein overall signaling. This is just an overview. We're going to go more in that. So we know that the signaling process is initiated by ligand binding to the receptor. Now, the receptor is now linked to the G proteins that are tethered in inner leaflet of the cellular membrane. Now, ligand binding to the receptor will result in interaction between receptor and G protein. And now this which will result in the activation of G protein. Now we know the G protein will now regulate the activity of membrane-bound enzyme that is found in the membrane. And the products of that enzyme activity are known as second messengers. So we see these second messengers will now amplify signal all the way into the cell. And also we see that they will be stimulated by ligand binding. So that is the process of signaling. So many second messages that we're going to talk about will activate a group of enzymes known as serine and threonine kinases. Now these enzymes will do phosphorylation of serine and threonine amino acids on certain proteins, affecting their activity. Again, these enzymes will phosphorylate the substrate on serine and threonine amino acids inside the proteins inside the cells. Certain proteins inside the cell will now get phosphorylated in serine, threonine amino acids. And this phosphorylation will change and alter the status of that molecule, of that substrate. And so this will result now in change of the activity of these proteins. Most of these targets or substrates or proteins are actually enzymes. So these enzymes, this phosphorylation will result in a change in the activity of this enzyme. So let's talk about heterotrimeric G protein signaling receptors. We know that large number of hormones and neurotransmitters have receptors on their target cells, and large number of them are linked to G proteins. Please understand that G protein signaling is the most abundant type of signaling in the body. That's why it is important. A lot of drugs, a lot of even diseases are associated with signaling through G protein. Now, when we look at the G protein receptor, so G protein signaling receptor in the membrane, common board question, is going to contain two domains. It's going to contain extracellular hormone binding region outside where it will bind to the hormone, and it's going to have intracellular region inside the cell that will react with G protein. Now, whenever they react with G protein, this will now evoke a secondary response, secondary signals, which will now result in the change and alter the activity of the cell. Now, please change this. You have an error on your slide, and it says that G protein, actually G protein receptors, are a transmembrane protein. So please understand, G proteins are embedded in the inner leaflet of your membrane, while G protein receptors are transmembrane proteins. So they cross the membrane in this several times. They usually consist of 7 membrane spanning regions. And these are most commonly have these 7 helixes that were connected in the membrane. Important thing for this is you're going to find a ton of these G protein receptors in the brain. Around 90% of G protein receptors in the body you will find in the brain. They're very, very active. So let's look at this. Let's look at how does the G protein signaling works. I'm going to use an example of a special subgroup of G protein called G alpha S. So GS. What does the GS stands for? S means stimulation. So which means that this system and this signal will stimulate something in the cell. It's not going to inhibit things, it's going to stimulate things. So what happens is you have unoccupied G-link receptor that will be in close connection but will not be bound to heterotrimeric G protein. So they're not bound to, they're close to each other and they're not bound and not interacting. So ligand binding will now bind from the outside to the receptor and change conformational state of the receptor, which will now cause interaction of this receptor with the heterotrimeric protein. So ligand binding will actually cause conformational change, again, that will enable an attachment of the receptor to G protein. Please remember that ligand molecules will always attach to a specific receptor. They won't attach wherever they want. They will attach with a specific receptor. That's really cool stuff. Ligand binding will cause now G alpha subunit, you know there are 3 subunits, alpha, beta, and gamma. G alpha subunit to release GTP, GDP, and attach GTP. That will activate G alpha unit. Now, what's going to happen? The G alpha unit is going to say bye-bye to its brothers and sisters. So it's going to go away. It's going to detach from beta and gamma subunits. And these free alpha subunits will now interact with an enzyme on the membrane that is known as adenylyl cyclase. Adenylyl cyclase or adenylate cyclase will convert ATP now into the cyclic AMP. And this cyclic AMP is now your secondary messenger that will going to go down into the cell, start activating different enzymes, and start sending signals down the cell. So this is a very, very common way of activation. Now, what's going to happen when the signaling stops? So when the signaling stops, what's going to happen is the hormone is no longer attached to receptor. It's gone. Receptor changes its conformational shape and detaches from rest of the G units, okay? G alpha, what's going to do? G alpha is now going to destroy GTP that is bound to it, and is going to make it into GDP, releasing phosphate. And it will go back and attach back to its 2 brothers and sisters. So it's going to go back and attach to beta and gamma subunit, and everything is done. No more signaling. Adenyl cyclase is not anymore stimulated because G alpha is not stimulating it. Signaling stops. So that is process of inhibition or termination of the signal. Now, there are various members of heterotrimeric G protein family. What does that mean? This slide just tells you there are tons of different alphas, betas, and gammas. So they will combine. You will get tons of different heterotrimeric G proteins because, just for example, there are at least 15 members of alpha subunits. So there are different subunits that are involved. So combination of various alpha, beta, and gamma subunits will form these heterotrimeric subunits. And we know that GDP, remember, is bound to alpha subunit of G protein. And whenever GDP is attached to alpha subunit, it's inactive. Alpha subunit is inactive, and it attaches to its beta and gamma subunits. Now, certain G-alpha subunits will interact with certain enzymes. So let me just explain the major subtypes of G-hemeric G proteins will have G-alpha S, G-alpha I, and G-alpha Q. So this here is alpha S stimulating. G-alpha I subunit, when activated, will inhibit adenyl cyclase. It's going to work opposite. G-alpha Q will activate another enzyme, not adenyl cyclase. It's going to activate phospholipase C. So that's important to understand, is there are different subunits. So identity of the enzyme determines which second messenger will be produced. But please remember, two main enzymes are regulated with G proteins, and these are adenyl cyclase and phospholipase C. So we're going to talk about these two. So let's look at the adenyl cyclase. So there are two different G-alpha proteins that regulate adenyl cyclase. G-alpha S stimulates it, G-alpha I inhibits it. So that's how they work. So a good example is, for example, epinephrine or adrenaline will stimulate your cells in your body. Why? By activating G-alpha S subunits in tissues. And what's going to result in the adenyl cyclase? It's going to get activated, and adenyl cyclase will produce cyclic KMP that will send signals in the cell. So epinephrine receptors that have large amount of G-alpha S are actually found in liver, muscle, and adipose tissue. And stimulation of these, I just want to show you where all this signaling goes, will result actually in a breakdown of stored carbohydrates, glycogen, and fat, which will be used as energy. So this signaling through epinephrine, through G-alpha S, will make cells break down their carbohydrates and fat and get some energy, release some energy. Glucagon is another hormone that works through this process. You're going to hear a lot about it, and will stimulate glycogen breakdown in liver, releasing glucose in the blood. So let's go and look at what's happening with G-alpha S. We talked about it. We have attachment to the receptor. Receptor changes conformation. We talked about it. Then G-alpha S will now release GTP and bind to GDP, GDP and bind to GTP, and then it will activate adenyl cyclase. So G-alpha S will stimulate adenyl cyclase, and adenyl cyclase will break down ATP into second messenger cyclic AMP. Now what cyclic AMP does is activate another group of cyclic AMP-dependent proteins that are known as protein kinase A, pKa. That's the next step in the signaling. How does that happen? Actually, cyclic AMP will bind to a protein kinase A, and will bind to regulatory subunit of protein kinase A, which will now release active catabolic unit that will be involved in the activity of this enzyme. That's the one that is catabolic. That's the real one that produces effect of this enzyme. Now, interestingly, when there is an increase in cyclic KMP, it's going to be automatically shut down. Cyclic KMP production lasts very shortly. Why? It's a signal. You don't want signal to be long, slow. You want signal to be boom, gone, boom, gone. Those are the signals. So what happens is cyclic KMP is rapidly destroyed by enzyme phosphodiesterase, which will convert cyclic KMP into inactive 5-adenosine monophosphate. So this will ensure very low levels of cyclic KMP in the cell. So let's talk about pKa. So what does protein kinase A does? Remember, it was activated by adenyl cyclase. So pKa will phosphorylate protein substrates, any other protein, other enzymes in the cell by working on serine and threonine residues. What does that mean? pKa will phosphorylate serine and threonine amino acids, making phosphorylated version in the proteins further down. This phosphorylation will now modulate the activity of that enzyme. It's going to make that enzyme work, or it's going to make the enzyme stop working. Okay? So that is the process. Now, this process of pKa phosphorylation of serine and threonine residues that goes further down inside the cell can be terminated by another enzyme known as protein phosphatase. It will stop signal. Signal is very brief. Phosphorylated, change activity on the enzyme, and stop working. Okay? And so protein phosphatase will cause dephosphorylation of those serine and threonine that were phosphorylated by pKa, and this will result in the termination of the signal. We know the G-alpha S will hydrolyze GTP into GDP, resulting now in the termination of adenyl cyclase activity and inactivation of the whole pathway. Now, let's talk about other group of G proteins. These are known as G-alpha I. They inhibit. So it's really intuitive when you look at this, is what would G-alpha I do? Totally opposite with G-alpha S. G-alpha I activation of G protein will actually result in the interaction with adenyl cyclase. However, when G-alpha I attaches to adenyl cyclase, it's going to stop adenyl cyclase from working. So adenyl cyclase will not work. So this interaction will lead to inhibition of the enzyme activity, decrease in cyclic AMP. Decreased production of cyclic AMP, of course, leads to decreased activation of protein kinase A, and decreased phosphorylation of serine and threonine residues on tissue, or in further down the road enzymes and proteins. So the whole cycling stops. That's why Dr. D wants to show you a clinical example. So the clinical example is cholera. I actually was misfortunate enough to see cholera epidemics when I worked for UN, and actually the cholera is a very serious condition. Actually has high mortality. The problem with cholera is this: it's actually acute infection, disease that is caused by bacterium Vibrio cholerae. And did you hear this from Dr. Kuo yet? Not yet, you will. It's a really nasty little bugger that looks like little commas that floats throughout the feces. Not very interesting, not very appetizing, okay? But the thing is this. This organism enters the GI system through food and water sources. So it goes from food, usually fecally contaminated food and water from patients who have it. It will result in painless and watery diarrhea. You're going to say, yeah, whatever, Dr. D, painless, fine. You know, I have a diarrhea at least, my tummy doesn't hurt. Yeah, but what happens is people will lose liters and liters of fluid. They'll literally lose most of their fluid volume through the diarrhea. And they will die because of hypovolemia. So that's the condition. It's actually reportable. If you ever see it somewhere, you're going to have to report it to the World Health Organization and establish a quarantine procedures, and you're going to talk about it more in infectious disease. So it's a serious condition. So what causes it? See, this is a Vibrio cholerae. Much nicer picture than what you would see if you stain it and look at from the stool. This is electron scanning microscope. But what it is with this condition is that it's not caused by bacteria itself. Bacteria is not a big deal. You get rid of it. Problem is a toxin that bacteria produces. Bacterial toxin is known as cholera toxin. And how does the cholera toxin work? It does this, guys. It modifies G-alpha subunits in intestinal cells. So these G-alpha S subunits cannot hydrolyze GTP. So what does that mean when they cannot get rid of the GTP? They remain active. You know, because they need to hydrolyze GTP into GDP to become inactive. So they're constantly active. So you have activated constant G-alpha S signaling, which will now result in increased secretion of chloride into the intestine. Cyclic AMP, high levels, will stimulate secretion of chloride through special channels known as cystic fibrosis transmembrane regulator channels. Don't worry, I'm not going to ask you those, but we're going to talk about them a lot in GI Phys. So they will actually cause a lot of chloride to go out. What's going to follow chloride is water. So this is going to be a problem. So the problem will be uncontrolled excess outflow of chloride out of the intestines, loss of fluid from blood. It's a fatal condition, not always, but in most cases, unless it is treated. How do you treat it? Antibiotics? No. Why? Because it's not a problem bacteria, it's a problem of toxin. So what you do with these patients, you actually have to rehydrate them, and rehydrate them, and rehydrate them, and on top of it, rehydrate them, okay? And what happens that the disease will take its course. Eventually, body will start producing antibodies against those bacterial toxin, that cholera toxin, and they will get better. Another condition is known as whooping cough. I want to show you two examples why these guys are important. Whooping cough does similar thing. Now, do the kids with whooping cough have diarrhea? No, okay? So we're talking about lungs now, but similar pathway. It's a highly contagious disease caused by bacteria Bordetella pertussis. It affects 16 million people worldwide. It's a really serious problem. And disease will start with symptoms very similar to common cold, and then these individuals will suffer from weeks and weeks of severe cough fits, okay? And the problem is it's associated with this. It's actually characterized by the presence of coughing that will be followed by this high-pitched whoop sound, like gasp after coughing. This can be associated with vomiting and fainting. It can be really bad. It may last for 100 days after the initial presentation. So think about it. You're going through this for a long time. What's the problem? Your patients will actually cough so badly that their capillaries in their eyes will burst. So they're going to develop usually conjunctival hemorrhages. They can pop out hernias all over the body because it increased pressure, specifically inguinal hernias. They can develop urinary incontinence, not fun. When they cough, they will actually pee themselves. And they can develop rib fractures. They can actually, because of the coughing, or actually even, they can cause dissection of their vertebral arteries in their back. It's a serious condition. Why it is serious? My previous school, we had an epidemic of this disease among my students. Why? Because they were sent in the area of the country where people chose not to vaccinate their kids, because it causes autism. So what happened is our students didn't get any boosters, so our students helped all these kids around, and they ended up getting a whooping cough, okay? So that is, and then it was so much fun to see a bunch of students whooping throughout the lecture, okay? But it's really, really not pleasant. So this is it. So children, more important is this, that this coughing can be induced by yawing, by eating, by laughing, and children under the age of 1, that is for all of you interested in pediatrics, will not cough. They will just cease breathing. And that is scary. They will cease breathing, and then, since they're small, you're not going to hear a whoop, but they probably have this condition. What causes this condition? Again, G protein signaling. Bordetella pertussis produces a pertussis toxin. And we can actually vaccinate against that toxin. We can vaccinate against cholera, too. So what is it? It produces a toxin that affects epithelial cells of the lungs. What does it do? It modifies G-alpha-I subunit that cannot inhibit adenyl cyclase. So now we're having a problem. We have inhibition of inhibitor, okay? The inhibitor doesn't work. So this will result in large concentration of cyclic AMP in their epithelium of their lungs, which will now cause excessive coughing, vomiting, dehydration. Treatment now is antibiotics. These guys you can treat not for a long time, but you do. Hydration, it's very common disease in third world countries and very dangerous. It is a rising disease in the United States of America. We thought we got rid of it. Why? Again, we have problems with vaccination. So everybody has their own personal opinion, but I get really angry when I talk about vaccination. Specifically by my friends posting all these ideas, I'm not vaccinating your kids. We're all doctors. We're going to practice in the future. We are here in medicine. We are here because of vaccines. If we never vaccinated our population, then probably half of the human population will be dead by now, okay? So we look at that as a very serious public health effect. So if we have this movement against public health, most powerful public health tool, where are we going to go? Yes, we're going to weed out our population easily, and you'll be dealing with diseases that you thought you will never see. Smallpox, things like that. Why? Because some patient says, because, what was it, McCarthy, or that famous model? She said that it caused autism. Okay, there are some things that are connected to these vaccinations, but probably, I presume, not autism, and never proven. Except the crooked colleague, research colleague of mine that I don't know, thank God, from Malaysia, fudged study reports and then suddenly reported and later admitted he fudged study reports. So it's going to give him a good publication. So that is the problem. Now let's go further. I can go on that for hours. But this is it. Let's talk about another enzyme that can be activated by heterotrimeric G protein signaling, and this is phospholipase Z. Phospholipase Z can be activated by presence of another type of G protein, GQ. We talked about G alpha I, G alpha S. This is GQ. It's like that awesome magazine, GQ. Okay, so Dr. Tolentino always looks like from GQ. He's the best dressed guy here, and I'm so jealous. So, but I'll get those suits one day. But GQ is actually, he does look good in it, so it's fine. Some people just can't wear tight suits, as they did. But so this is it. So hormones by GQ linked. So hormone will bind to a GQ linked receptor. And what's going to happen? Same story. We're just putting Q instead of alpha. Okay. The hormone binds to the receptor. Receptor changes its conformation and attaches to heterotrimeric G protein that has GQ in it. We know the GQ attaches first to GDP. It's going to drop the GDP and take GTP and become active. Whole story, same. But what it's going to now do is that alpha subunit will activate the enzyme known as phospholipase C. Now, this is a little bit more complex, but you learn it. Is that phospholipase C will cleave now another membrane protein known as phosphatidylinositol 4,5-biphosphate, or PIP2. So when it cleaves it, it will cleave it into the inositol 3-phosphate, IP3. And this inositol 3-phosphate that is cleaved now will be sent into the cytoplasm. It will also break down the phosphoinositol diphosphate, or biphosphate, into diacylglycerol. So it will cleave it phosphoinositol diphosphate into inositol 3-phosphate and diacylglycerol. Diacylglycerol stays inside the membrane. Inositol 3-phosphate goes into the cell. Inositol 3-phosphate will now travel inside the cell. So what's going to happen with inositol 3-phosphate? It's going to bind on a specific receptor on the sarcoplasmic reticulum of the cells. So what does the sarcoplasmic reticulum of the cell contain? It contains calcium. So the calcium will be released. So what do we have? We have 3 secondary messengers, do we? We have diacylglycerol. We have phosphoinositol 3-phosphate, IP3, and we have calcium. All these 3 are now secondary messengers. Now what happens is that this specifically calcium and diacylglycerol, not phosphoinositol 3-phosphate, will now stimulate an enzyme known as protein kinase C, not protein kinase A. And what protein kinase A is going to go? It's going to go further down, and it's going to cause phosphorylation of tons of specific amino acids in further down pathway. So that is the characteristic of this. This is a very important pathway in your body. Let's talk about another group of signaling. This is known as RAS-G signaling. You remember the Rasta guy I mentioned at the beginning, Jamaica? Okay? So this is a different signaling. It's still a G protein signaling. However, this is not it doesn't have 3 subunits. It only has 1 unit. Okay? This is RAS-G. So RAS-G proteins are actually very similar to the alpha subunits of heterotrimeric G protein. They do not regulate membrane-bound enzymes. So they do not touch upon membrane-bound enzymes. They do really cool stuff, and I'll show you in a moment. And they do not result in a production of second messenger directly. But they still signal. How do they do this? Activation of this process, RAS-G, will activate phosphorylation cascade. Think about it. You activate RAS-G, and when you activate it, there's going to be phosphorylation cascade. One protein will phosphorylate one, and another will phosphorylate another, another will phosphorylate another, another will phosphorylate another, all the way down to the nucleus. Okay? So and then this protein that is phosphorylated in the nucleus will regulate transcription of genes. It's kind of cool. So it actually is involved in regulation of cell proliferation or multiplication. So this RAS-G protein act as relay switches between the surface receptors from outside and the DNA transcription, all the way down in the bottom. So that's their important situation. What's the problem? If the RAS-G protein's signaling is not working properly, what's going to happen? This can cause an development of cancer. So I'm going to show you that why in a second. So this is how the whole signaling happens. Now we're talking about a special type of receptor. We're not talking about the G receptor. We're talking about something called a catalytic receptor. If you understand this receptor now, your next lecture will be breeze, okay? So this is a catalytic receptor. It's a different type of receptor that works with good old RAS, okay? It's not the same receptor, the 7-membrane spanning receptor you've seen that works with G. This is a different receptor. So ligand will bind to catalytic receptor, which will cause now the receptor to phosphorylate tyrosine residues in his cytosolic end. So think about it. Something attaches from the outside to the receptor, and receptor in his tail inside the cell will phosphorylate its tyrosine residues, tyrosine amino acids. So it's going to light up like a beacon. Boom. It gets phosphorylated tyrosine. Okay? It has phosphotyrosine. Now what happens is, this now phosphorylated tyrosine will act as a beacon and start the signal. Now the receptor of phosphotyrosine here will now act as binding sites for something called intracellular adapter proteins, proteins that will adapt the signal. And in this situation, the next one we're going to mention is SHC, or in your image, GRB2. SHC is the adapter protein. Now, adapter protein will have on its a domain known as SH2 that enables adapter protein to bind to the phosphorylated tyrosine. Now, what's going to happen now is next guy is going to join the party, or a gal, and it is another adapter protein known as SOS. Just remember that now. SOS comes into the party. It's another adapter protein. Now, SOS binding will result now for good old RAS to join the party. Now RAS is going to say, ooh, there's 3 of you, I'm coming. So what's happening is, you know, that you have SHG, SHC, you have SOS, and now RAS binding all together. Now this whole shebang is happening. Why? So the RAS will release GDP and attach to GTP. Okay? So that is the problem of it. So it will actually exchange GDP for GTP on RAS. Okay? So that is going to activate our good old RAS. Now RAS GTP will promote now binding of another guy. There is a new party. There is a new Rastafarian joining. And this guy is known as RAF. Okay? So the RAF is actually a serine protein kinase. Who is RAF? RAF is known as MAP kinase. Okay? Kind of cool. So it's mitogen activated protein kinase. Okay? So what does it do? It activates all these further down. Now MAP kinase will phosphorylate and activate who? MAP kinase. Okay? One less. Okay? So now the MAP kinase is going to now activate whom? MAP kinase. We're left with, we're left with just one kinase, okay? So that is actually known as MAPK, or MAP, also known as ERK. And this guy, this MAP kinase or ERK, will travel all the way into the nucleus, and what it's going to do is going to phosphorylate and activate factors that regulate gene transcription. So now we're regulating gene transcription. So this cascade will result in activation of transcription of genes involved in cell division or cell multiplication. How this is all going to stop? It's going to stop as GTP is hydrolyzed by RAS and becomes GDP, and RAS gets away from that party, whole thing falls apart. Why is this important for you as physicians? There's going to be a problem. You might have situations where this RAS mutation will occur. There's going to be a problem with the RAS signal. What does that? Is look at this. Some mutations of RAS, So you have an individual, some cell that has mutation of RAS. Okay? Now, what does that mutation will do? It will prevent RAS from getting rid of GTP and putting it into the GDP. So it will inhibit RAS of inactivating itself. Make sense? Because it needs to convert GTP into GDP in order to inactivate. It cannot do that. It's weird looking. It doesn't work right. Okay? So it's going to constantly remain active. Okay? So result will be conversion of GTP to GDP. We know inhibits the signaling. But if there is no conversion of GTP to GDP, signaling is constant. There is a constant signaling. What is this a problem, guys? Constant signaling through RAS will lead to constant multiplication of the cell. What does that mean? Cell will divide and divide and divide and divide and divide and divide and divide when it should stop. And as it divides, cell will start producing cells that have eyes on top of their heads and noses on their back. Okay? It's going to start producing abnormal cells. Mutations start accumulating. Cells become cancerous. Okay? So constant stimulation and uncontrolled, we're going to talk about it tomorrow, uncontrollable replication will lead to cancer. Now, so that is one of the reasons why this is important. Now, why is this important? It's important for the condition we're going to mention is neurofibromatosis. This is a condition that actually, I always go to my stories I diagnosed first time for my woman that used to cut my hair in Virginia. Okay, she just kind of said, "Oh, you're a doctor." I said, "Yes, you teach in the med school." Yeah, I have all these things hanging, like start popping up on my back. It's like neurofibromatosis, 100%. So this is actually a condition. It's condition that's associated with the problem of RAS-G protein signaling. It's a genetic, it's inherited condition. So this neurofibromatosis is a number of inherited conditions that are clinically and genetically distinct and carry high risk of tumor formations, specifically in the brain. These individuals will develop tumors in the brain. So this guy you see has neurofibromatosis type 1. These individuals will have tendency to produce large amount of nerve tissue tumors. These are all nerve tissue tumors. Usually don't hurt, okay? But what happens is that and interestingly, I don't know if it's good news or bad news, but probably a good news, is they don't become cancers. But the problem is that they will disfigure body. They will push structures around themselves. They will actually make serious problems. So they may cause damage by compressing nerves and other tissues. The next is neurofibromatosis type 2. Neurofibromatosis type 2 is a similar condition, but what does it do? It causes formation of bilateral acoustic neuromas on both sides. What are the acoustic neuromas? These are the tumors of nerves. So people with neurofibromatosis type 2 usually don't have skin issue. They will have other issue. They will develop bilateral acoustic neuromas. They will develop tumors of their vestibulocochlear nerve, cranial nerve 8, which ensures that you hear things, ensures you keep your balance. So, but specifically, it will affect the hearing. So these individuals with neurofibromatosis type 2 will usually lose their hearing later. The next thing is schwannomatosis. It's another expression of this process, and it actually is characterized by formation of schwannomas. Schwannomas are neuronal tumors of the neuronal sheet, not neuronal cells, and they're very painful. When they occur, if you press on it, people will hurt because you compress the nerve under it. And these individuals who have this type of neurofibromatosis that is known as schwannomatosis, sadly it sounds like a sandwich you buy, like schwannomatosis. Come on, guys, jokes, okay? But, give me 2 schwannomas, please. No, actually, don't use that joke. But, but it's a good one, come on. But the schwannomatosis is actually a condition. So, why I'm joking about it, I can say it's going to be recorded. I'll probably end up in a divorce. My wife had a schwannoma. So, in her roof of her mouth, and I talk about it as a very glorious moment of our lives because, all her life she had, interestingly, a speech impediment in our language. Never in English, interestingly. So, so that tells you, how hard it is to speak English. I didn't say that, but but we have a lot of R's, so she had problem producing R's. So for years, everything fine. Okay, she talks that way funny. It's actually cute. But yeah, it is, but she doesn't speak. She speaks in English with me now. But the thing is, she went for surgery and they found a tumor on her hard palate. And, I knew immediately tumors on hard palate, 90% malignant. So, when I found that out, she was in Wake Forest a long time ago. She's still just getting into her residency. She was like, oh my God, you went through all this and you're not going to pay off your student loans. I'm just joking. But what it is, and there's a whole story. Eventually, they found out it was schwannoma. So actually, she had just one schwannoma actually so it doesn't mean that it has schwannomatosis, no. But the thing was, they removed it, and and I was like, now you're going to be able to speak in our language. No, she still has that cute thing. It's kind of cool. But but that was kind of cool. So schwannomas are not only associated with schwannomatosis. You'll see it in some other conditions. Some people might have it somewhere, and under skin. Now, this isn't what causes neurofibromatosis. This is a sad situation. This is actually an individual who has neurofibromatosis type 1. You see that those tumors will seriously grow and they will actually deform, disfigure. And this disease is characterized by the absence or abnormal production of protein neurofibromin. I'll tell you why this protein is important. This neurofibromin protein that should be normal, and these individuals, they do not produce normal neurofibromin. So actually, its production in normal situations is encoded by NF1 gene, neurofibromatosis 1 gene. Easy to remember, okay? And it actually, this is very common board question too. So neurofibromatosis 1 gene will encode for the production of neurofibromin. And what does the neurofibromin do? It's known as a tumor suppressor protein. And we're going to talk about it tomorrow. These are the proteins that prevent your cells becoming cancerous. Okay? They work for you. They prevent tumor formation. Now, what's going to happen if you produce? So how does it achieve that? By inactivating RAS, by inactivating abnormal production. Okay? So what's going to happen in these situations? If you have mutation of neurofibromatosis 1 gene, you will have decreased suppression of RAS activation. So RAS will now be activated more than it should be, and they will develop problems. And hyperactive RAS will lead to uncontrolled cell growth and division. So that is it. Now, I just want to kind of show you. This is a kind of example of one clinical case, briefly, and I'll let you go. This just shows you a 7-year-old male. I kind of modified a case I found online, and I want you just, when you have time, just read through it, because it is very common. This is what you're going to be working with, is whooping cough. Usually, you're going to have a kid that was not vaccinated, but don't think that you cannot get whooping cough if you were vaccinated. You have to get a booster, okay? So that's why it's very important when you become, physician active and even now when you work with patients, vaccinate yourself, They say, you can't help others if you do not help yourself, okay? So make sure to do this. Again, I remember in Virginia, that was really interesting situation. I had like 20, 30 students who ended up all getting whooping cough because going up there. Actually in the area known Floyd, which is an awesome area, but area where parents usually, don't vaccinate their kids. Very hippie area, so they want natural stuff, So they were helping them with teas, things like that. So just be aware of it, that it is on the rise. Okay, guys, I'll give you a little break, and we'll continue back. Thank you.